Okay, we are recording. Are you ready, Dr. Hudson?
Yes, or you're going to talk about some things first, or you want me to talk about my things first?
I was asking if you're ready, if we got the meeting started. Like, I'll talk about some stuff beforehand, and then we'll get to your presentation.
The only thing I'm wondering, okay, I had my PowerPoint ready here, and now I'm thinking, okay, now I don't see it anymore. I don't know if maybe if somebody goes to screen share, maybe I can find it. I must say I'm a little bit, yeah, there we go. So there's my PowerPoint that I'm going to do. I don't know if you can see that. If we share that screen or how I pull that up.
So maybe at the bottom of your screen, there might be a little button where it says share screen.
Yeah.
Click on it and see what happens.
Okay.
And then I think you might be able to pick like what screen. Yeah, there you go.
Is it there?
Yes.
Yeah.
Yeah.
Okay. Well, and whenever I would go, and you guys can maybe help me, if I go to like slideshow from the beginning, it takes the whole screen and I can't see anybody, but then maybe I don't need to see anybody. So do you guys want me to kind of do my 30-minute, 20-minute? I don't want to keep y'all all night. And that way, or do you want to talk about your meeting first?
If you wouldn't mind, if we could talk about the meeting first, and then we can get a presentation.
Okay. Should I leave this up, or should I, your screen sharing, should I stop the share and then try to start it up again, or what do you think? Or I'll stop the share?
Morgan just took care of it.
I fixed it. You're good. So you can see exactly where you are, and I think whenever I stop, it'll pull yours back up. So you should be good.
Okay, well, welcome back, Tribeta members. We are going to kick off the spring 2021 semester like this. We're going to do it over Zoom. We're going to continue it over Zoom as long as the pandemic's going. But other than that, just welcome back. Glad to see some familiar faces, or those faces that I can't see. Okay, next slide, please, Morgan. Oh, can you go back some? There you go. Okay, just a few housekeeping details to remember. For at least the Delta Tau chapter, Dr. Harper requires that we do 12 hours, and those 12 hours can consist of the meetings, which we do require 6 out of the 7 meetings for all members to attend. Other than that, you can get hours with multiple ways that we can go, that we'll talk about later in the PowerPoint, and just some other general stuff. We will have officer elections and new member inductions at the end of the semester, but that's also a ways off. So just a little preview before that. Next page, please. So typically, oh, Jennifer left. Okay, so typically we would have trivia.
Are we looking at the right slides?
Are y'all not looking?
So can everybody see the Tribeta slide?
No.
Well, makes a whole lot of sense now. Okay. Hold on. Let me see what happens.
So I think what you have to do, what I had to do, Morgan, is everyone has to go up to view options on the top of that screen, and then click Morgan's name. It has Dr. Hudson's name and then Morgan's name.
Oh, I didn't even know that was a thing.
Oh, that's cool. Well, I learned something new.
I'm on the phone, so it does it automatically.
Whoops.
Okay. So can everyone see the PowerPoints with like the housekeeping details?
I can't see them.
Okay.
Can't see them either.
Okay, so Dr. Hudson, I'm gonna have you stop sharing your screen, and then maybe it'll pull mine back up.
Yes.
Yeah, that worked.
Okay, good deal. All right, Morgan, would you go ahead and go to the trivia night? Thank you. Okay, so again, we typically have it in the spring semester. However, because of COVID, it's not exactly suggested that we have any get-togethers. Also, it'd be a hassle to do that with the university, as we're required to do some events. we are required to schedule large events through the university to get their okay for it, just as a liability stuff. So instead, we'll just host trivia night over Zoom, and we'll do that with Kahoot. You know, everyone loves Kahoot, at least what I've heard. I haven't heard a single person really say, "Oh, I hate Kahoot." So we'll just do that. It'll be a fun, free event for anyone to enjoy. So if any of y'all, any members who want to join, or get their friends to join who want to like try to flex how much biology they think they know, please encourage them to come along. But we'll do that later in the semester, more than likely closer to finals. Sorry for that. Okay. And so ideas for hours that y'all can get, obviously the meeting. So in order to remain an active member, you'll have to, sorry, I'm spacing out. Have to attend 6 of the 7 meetings. You can obviously attend all 7 if you want to. Attend trivia night. That will be more than likely. We'll give y'all 3 hours for that one. And we also have the South Central Regional Tribeta Convention, which the date has not been confirmed by the organizers yet, but it is in the works. And you can also find that on the Tribeta or the main Tribeta website. As far as like other hours through like volunteering, we're trying to set up tutoring with the high school. Alicia graduated, so all of the information that was with her is still with her. I'm still trying to contact her in order to get all the information and the contact from the high school so we can actually get that up and running for y'all. Trash pickup. I don't know if we're going to be able to do a state park cleanup like we've done a long time ago. I don't think I've ever been in one of those. That's how long ago it's been. But we could talk with the state parks. We could also talk with the school about that. In addition, you could one trash pickup that we did last semester that was completely on your own was pretty much just grab like a Walmart bag and just go out in like the woods or around Huntsville and pick up pieces of trash and essentially fill up a trash bag, send us a picture of that, and then we'll give you 1 hour. It's super easy. I know I filled up 1 trash bag in like 15 minutes. So, I mean, it's really not hard. Also, the Sam Houston Museum of Natural Science, they're always looking for volunteers. They've typically got or they're typically just missing enough volunteers to get a lot of their projects running. But if y'all want to do that, it's a really easy job for volunteering hours. Also, it's just you get going to the museum, you start doing stuff, and you get to enjoy the nice museum smell. But we're also going to have habitat restoration potentially and Dr. Hudson will talk about that. And did the screen stop sharing?
My connection flickered. Let me fix it.
Yeah, well, I know that's the last page before Dr. Hudson. So, and will also encourage members to do blood drives. So as long as you get a signed sheet by like the blood drive people, just basically show proof that you were there, we'll give you some hours for that. And then without further ado, our speaker for tonight is Dr. Hudson.
Okay, let me see if I can do screen sharing again. Okay, and I'm going to see if I can put it on the slideshow. There we go. Okay, so that looks like it's working. So I'm Dr. Hudson, and I've been in the biology department longer than anybody else. So that's probably my kind of claim to fame here is being here. I came in 1991, so way before most of you were all born. So been here a long time. And my research organism is a fern gametophyte. So when you took botany or if you took Bio 1 somewhere, Bio 2, you might have learned about the fern life cycle. So I was going to work on that first. But before I get into that, I'd just like to mention that Tyler had mentioned about the habitat restoration. That's at the field station. And I don't have any pictures of that. But what we were going to hopefully meet this Saturday. But turned out I need to fill out one of those forms and the university need the President's Cabinet needs to approve what we do, our work days. I don't call them an event, I call them a work day. And anyway, we can't meet this Saturday because the form hasn't been approved yet, but it's supposed to be approved next week. And what I'll do is I'll let everybody know, and Tribeta members are very welcome to come out there. You want to wear your old clothes and old shoes. And what we do is we clear understory. If you've been in ecology, we've cleared understory trees and shrubs. Alan Bybooth, the field station manager, he cuts down a tree, and then we kind of swarm on it and cut it into smaller parts and haul it away, or haul it onto a big pile, or put it in the wood chipper. And we're trying to prepare a habitat for the red cockaded woodpecker. It had been, or it maybe still is considered endangered, but there's been some, suggestions that maybe it should be just moved to threatened. But nonetheless, 30 years ago, there was a colony out at the field station, and then the habitat kind of changed. Prescribed burns are very important to keep the vegetation down, the understory down, and that really wasn't happening. So the understory grew, and the red cockaded woodpecker either died off or left. So we're hoping to restore it. It's going to take several years, but it takes a lot of manpower, physical labor. You can't use a bulldozer or front end loader or something like that to do it. You've got to really just get people working by hand, and that's where we've made some progress. In fact, we made quite a bit of progress, but I think Alan said 11 acres cleared, and we still have a ways to go. So Tribeta, over the last 3 years that we've been doing it, has really stepped forward and come out and helped us. So when we have a work day, it's usually Saturday between 9 and 12, or we're going to have some on Fridays between 2 and 5. And so Tribeta members are welcome. It's a nice, fun way. We can do social distancing because we're out in the woods. And I have gloves that I bring for everybody, and then we put them in a bucket and I wash them. If you've got some goggles you want to wear, otherwise I've got some goggles, and we'll put those in a bucket then too after people get done. But anyway, it's pretty much fun. And I don't know, we do that, but that's another way. If anybody wants those, get your volunteer hours, that's a good way. And Tyler's been coming. He knows what's going on out there. So if you need more information, you can ask him. But we will not meet this Saturday because I don't have permission yet. And I should be given permission next week. So hopefully the President's Cabinet will approve my application. But getting on to what I do for research, things maybe I've done in the past, things I'm still interested in, hopefully we'll do. If we take a look at the fern life cycle, I don't know if I can, I'm not going to try to do anything with the thing. If you look over there on the left, you can see the fern that everybody talks about. Yeah, that's a nice, I like ferns, but what I'm more interested in is what's over on the right-hand side, what the little spores grow into. And so you'll see some pictures of gametophytes. Those are those gametophytes, little heart-shaped, green. They're photosynthetic. They're on their own. And then with fertilization and in the presence of water, you get the sporophyte generation. So some of you might remember that from botany, but I know you guys have a lot to remember from botany. So anyway, what I'm working on is the gametophyte. Let's see if I can advance this. So one thing, because I've been working with gametophytes since, 77? So fern gametophytes since 1977. And so I've been there, just like you get kind of a research bug, and but it's not a real bug, but I got the fern gametophyte bug. And so that's what I'm primarily interested in. And so one thing is I've been collecting fern spores of this one particular fern that I've worked on for all these years, and it's called Anemia mexicana, grows in the hill country. So if you're familiar with the hill country, kind of west of San Antonio, that's where it grows. Also, obviously, into Mexico. but I have spores that are stored for, since I think the earliest ones are 1978. And so I've put them in the refrigerator. I was going to throw them out because I thought surely they're not viable. But lo and behold, I put them on some auger plates and they started growing. So I thought, well, let me do a study and see if they, what the viability is. Are they growing like they should? And so that's where because I have spores of different ages stored in the refrigerator that I never used, I did this study. I should mention that for fern spores, there are some fern spores that are green and they have a chloroplast. They have short viability, maybe 60 days, equisetum, anoclea, and there's others that are that way. But the spores of anemia and many of the ferns here in Walker County, they are not green. They have stored proteins, lipids, a thick spore coat. So they are not green. But people say, oh, they're viable for a while, but nobody's really studied them because you can't study them unless you've had them sitting in a refrigerator or sitting somewhere for a while. And that's where one advantage of studying them for so long and not throwing anything away, they're there. So this is Anemia mexicana from Real County out in the Hill Country. This is just one leaf. And what makes anemia different from other ferns is that it's a pinnately compound leaf, but the two bottom pinnae are where the spores are located. So they're not on the underside of the leaves. Usually you'll see me, if I'm out at the field station, I see a fern, I look on the underside of the leaf because that's where the sori are. But with this fern, you don't look on the underside of the leaf. It's got those two basal pinnae that have the spores. So it's really easy to collect the spores. On this fern, you don't even have to collect the whole leaf. And here's what some fern spores look like under the dissecting microscope. They're brown of anemia. They've got some ridges on them. We can see those. Here's one seen with the electron microscope. And just like pollen, so maybe some of you have heard pollen has different coats to it, outer coverings on that pollen, the same thing with ferns. So there's maybe, several thousand species of ferns. I'd have to go back over here. How many did I say? No, I guess it's not. Oh, 11,000. 11,000 species of ferns. When you look at the fern spores, every one of them's different. So just like pollen in the flowering plants, fern spores are different. This one happens to be Anemia mexicana. And if we look on the inside, this is a spore that has just started to germinate. It's split open. And it has, this one, in fact, is made up of 3 cells, but, and you can see the nucleus on those cells, but most of that spore is made up of that clear material, which is lipid, doesn't pick up any stains, and then some dark staining bodies, which are protein. So the spore, before it actually cracks open, when it's like that, you can't even fix it. You can't prepare it for microscopy because it is like, well, if we look here, that spore coat is really thick and it's impermeable to fixatives and materials used in, microscopy. But once it cracks open, then you can get fixatives. You can then start sectioning it and then stain it. And so this spore has already germinated. You can see it where it's open. And what's going to happen with anemia is that it's going to open on this triangle that you can see, and that's called the triradiate mark. And that's where it's going to split open and start growing. So I took spores, non-green spores of anemia, long viability. Some of them then I calculated 42 years. It turned out that viability was very good, but then I said, well, okay, viability, they germinated, but how do they grow? Do they grow as they're supposed to? So believe it or not, and I'm talking a little fast, but I don't want to keep you guys here all night either. So for fern spores of different species, they grow in slightly different ways. They don't all look alike. And I'm going to go through, maybe you shouldn't be recording this because I am really talking fast here. But to collect what I did and what had been done in the past for the past 42 years, I guess, was I collected spores and put them in paper envelopes where they dry and they're released from the sporangia. And then I took those spores and put them in glass vials, and I stored them in the refrigerator at 4C. I didn't store them in liquid nitrogen. I didn't store them in the freezer. I just stored them in the refrigerator. Should I have done them in the freezer? Maybe so. Should I have stored them in liquid nitrogen? Maybe so. But what this study was, and what I've had, is just refrigeration. And then we took agar solidified medium. If you've had micro, you have done a lot of, bacterial, those little plates with all that culture medium and bacteria. We use those same plates, but the medium that I use for fern spores to grow gametophytes, I don't need any carbohydrates. It's just minerals, because in your bacteria, unless they're blue-green bacteria, are going to need something other than light. They're going to need some kind of carbohydrate food source. For my fern spores, they don't need any carbohydrates. So it's just minerals, nitrogen, phosphorus, calcium, micronutrients, micro minerals, just copper, iron, very small amounts of some of that stuff. But in an agar medium, just like what you'd have in micro. And then we took the spores and just kind of dusted them very lightly onto the surface of the culture media, and then put them under fluorescent lights at room temperature. And after a few days, they started to grow. So I had, the earliest year I had was 1978, and then I had several other years in between up to 2019. And then photographs were taken with a dissecting microscope and a digital camera, but lately phones have been, the camera on a phone is almost as good as a digital camera that I've got. So we've been using, phones also. And what I found was variation exists within each year, but really no difference between the new and the 42-year-old spores. So there's variation that exists within 1 year, but, from new spores from a year ago in 2019 and spores that have been in the refrigerator for 42 years, not really much difference. I should point out too, is that these spores are really small and they are just single celled. If you look at the smallest seed that, you can think of, orchid seed, bromeliad seed, the mustard seed referred to in the Bible, those seeds are made up of lots of cells. These spores are one single cell and they are living, although they're primarily, a lot of lipid and protein, but they are, not much water, no water, or very, very, very little water. And that's characteristic of a seed also. But these spores, very dehydrated, mostly lipid and protein, but, it's got a nucleus, it's got mitochondria, it's got not chloroplast, but what we would call proplastids. So, and ER, all the, the parts of a cell. But spores are one single cell, whereas, seeds, even if the seed is small, it's still lots of cells. But let me show you some gametophytes. So here's a spore cracking open at 4 days. So it's just opening up and it's going to start dividing. And here they are under the dissecting microscope. So these are some spores at 7 days from 1978 collection. My graph on the bottom there is a ruler, and the distance between the two right there is on the bottom is a millimeter. So these things are little. We can't see them with the naked eye. We've got to have a dissecting microscope, and it works really, well. And you can see the gametophyte is growing on the plate. It sends out an elongate rhizoid, and then it has its green, what we call the prothallus. And here it is at 10 days. So now the cell's carrying out mitosis, getting bigger. It's making a flat little two-dimensional structure. This is where, if anyone, I'll just put it out there, if anybody's interested in looking at living cells. I worked with Raj a little bit with the confocal microscope. These are really nice to look at because they can stay alive, and they're one cell thick. So you don't have to section them. They're just naturally one cell thick at this stage right here. And I thought I had some pictures with the confocal, and I know they're somewhere on some file or some jump drive, but I couldn't find them. But anyway, they're really interesting. The cells are packed with chloroplast, and they're carrying out, some of them are carrying out mitosis. Here's at 12, you can see they don't all look exactly the same. And here's one that's developing what's called a lateral meristem. Remember, when you look at fern gametophytes, oftentimes people show it as that heart shape. Well, this particular fern gametophyte starts out as kind of a oblong heart, or kind of a asymmetrical heart, and then eventually it will become heart-shaped. But notice, and I don't have a pointer. I think was it? Can you guys see a pointer? You can?
We can see her.
So like right here is one big cell. Here's that lateral meristem right there. So it's a heart. It's not a heart yet, but if you watch development, and that's where something I've been interested in, development for years. So I'm not interested in just the finished project. I want to see what it happens as it goes from a spore to a mature gametophyte. Here now, these also have, you can see here's like a big cell, here's smaller cells. Right here is that lateral meristem again, and now it's making these little marginal trichomes. They've got trichome and other, little marginal hair. So it's and this is all two-dimensional. It's not it doesn't have thickness. It's only one cell thick. So they vary in size. Here, this one's making a little lateral meristem, varying in size and shape, but this is all within 1 year. And even the new ones are the same way. So their variation in size, smaller ones, bigger ones. This one's getting to look a little bit more like a heart. We can see a tiny little hair right here. One of their lateral trichomes. They're made, all the cell division of this thing to get bigger is happening right in here. If you wanted to study mitosis or look for mitosis, it's happening right here. These cells are they're made, they're carrying out photosynthesis, but all the cell division's happening right in there. Here's now one that it's becoming, sometimes it's hard to get a good picture if they sort of get a little wavy, but here's that lateral meristem, and you can see these nice cells. And this is, I've written here core date, that's 24 days. So it's a kind of not a real long-term project, but takes a little while. Antheridia, these two little things right here are the new forming Antheridia. So they form oftentimes right near the notch in this area that we call the midrib. So at 26 days, I can see some nice Antheridia. And by 31 days, all of these little spots right here, these are all Antheridia. And Antheridia are round, and they sit on the surface. They're on the surface of the gametophyte. So this is still very two-dimensional, no thickness over in here, little thickness maybe starting up here. And now we've got these Antheridia, they're superficial on the surface. Somebody might say, well, what about Archegonia? Some of the gametophytes at 31 days don't have any Antheridia, and those are the ones that are going to form Archegonia. So here's another one without our Antheridia, no Antheridia, you're going to be Archegonia. And this one's got a nice little trichome. Let's see if I can see it. There we go. That little clear little hair. It's characteristic of the Anemia mexicana, but it's not, it's sort of something that's unusual for this one. And then now here's another one that's a male, and that one's got a lot of Antheridia. There's a couple of trichomes there. Again, getting bigger, nice and heart shaped. But see, this is where if you don't look at development, you never realize that the heart was lopsided. during development. You'd think it was always a nice heart. It's like, no, it had a lateral meristem. Here's another one with antheridia. And then, oops, or you know what? Let me go back. I think this one, maybe this one doesn't have anything. This one has antheridia. This one's going to be female. This is 35 days. And here, it's really hard to see, but right here are a couple of archegonia. They're kind of poking up. And I can recognize them because I looked at them for all these years, but it would be hard to, if you're not used to looking at them. Here they are a little bit closer up. These are archegonia. They're not, there's only maybe 3 or 4, maybe 5 archegonia that it'll make, whereas with antheridia, usually there's a lot. Here's a big female gametophyte. Usually they're bigger in size with a couple of archegonia that are forming there. Usually they have a much thicker midrib also. Here's one with antheridia. So the antheridia look like they have some kind of disease, lots of antheridia on it, and just a few archegonia. And this is what they look like on the plate. So here's my agar solidified media, and we've got, thousands here. So what we've been looking at is just, 1 or 2, but there's a lot. Oops, let me go back here. So variation exists. So I concluded variation exists, different ages, but essentially no variation between the ages. So my 42-year-old spores are just as viable as and grow just as well as 1-year-old ones. If somebody would be interested in development and want to do a study, I have old spores of Palea atropurpurea, another fern. I have spores of Theliptrus kunthii and also Pteridium aquilinum that are probably 30 to 40 years old. And you might say, well, they're going to be exactly the same. Maybe they will, but maybe they won't. We got to do a study. So that's science. Something else I've been doing, and I'll tell you about this, is spore production in a couple of ferns that are out at the field station, Asplenium platyneuron and Woodsia obtusa. So I had some students, they really weren't too interested in looking at, things under the microscope quite growing like I was. So we did a study on spore production. And so here's a picture of Asplenium platyneuron. It's called the ebony spleenwort, grows out at the field station. And here is Woodsia obtusa. It's called the blunt-lobed clipped fern. Both of those out at the field station, so easy to get. How many spores are produced by a single frond? If you look in the literature, maybe about 10 different ferns of those 10,000, people will say they make lots. And you think, okay, well, how many is lots? 1 million, 1,000? How many spores do they make? And so, maybe for 10 or so species, people have said, oh, they make 1,000 or 10,000. Most of them just say lots. And then when you read the paper, it gives no directions on, how did they do it? How did they calculate this? So we set out to try to, figure out a way to give a pretty good estimate as opposed to just saying, oh, it makes a lot. And so we've collected ferns from the field station, collected fronds. We dried them in a plant press. And then we divide it, say one particular, one frond here, because they're not, all the little leaflets on it are not all exactly the same. We divided it into sections, 3 sections. So we had a top section, kind of a middle section, and then the bottom. And then we started counting in each section the number of leaflets per section, the number of sori per leaflet, number of sporangia per sorus. Remember, those sporangia are clustered together into a sorus. And then there are what calculation is, there's 48 spores per sporangium. So we counted spores per section and then eventually spores per frond. So here's the underside of one of the leaflets, and you can't hardly see it very well there, but there are individual, there we go. The sporangia or the sori are kind of elongate, and we can see these have released all of their spores already, but we can see this annulus. So we could take this, and what we found is if we put a little bit of immersion oil. So if you were in micro and you put your thing on immersion, on the immersion one, if we put a tiny little drop of immersion oil on here, we were able to count these things and kind of manipulate them a little bit. If you take these and they're dry and you try to manipulate them with a very fine pin, like a probe is even too big, but these little insect pins work pretty good. What you find is that they just start flying all over the place. So then you can't count them. So we use that immersion oil, and it worked really very well. It helped us. And so this was Woodsia, and now you can see a little bit better here. Here's a sporangium, here's a Saurus. We counted, came up with, averages, and we found, well, I'm going to pass over all of this and go to our last slide here. Oh, well, let's see. So we found, say, in this particular one, well, let's see. I think this was Woodsia here, different links. This is kind of covering up some of mine on here, but let me just move on, because we determined the number, and I've got a slide that says those numbers here. But the next question, oftentimes you guys will find, if you're working on a research project, you figure out something and then you ask another question. And we said, okay, so here we've got a plant with, 6 or 7 fronds or 8 fronds. They're making, all these spores. Do we have any spores growing into gametophytes? So the next question is, can we find gametophytes? Well, gametophytes are really small, but we looked and we were able to find little sporophytes starting to grow, and those little sporophytes would be coming from gametophytes. And then when we looked even closer, and you've got to pretty much put your nose to the ground, you could see some little, we were able to take some soil samples, put them under the microscope then. So we had this picture's with the microscope, can't see it very well, this detail, but we could see a young little sporophyte and then a little gametophyte. And we found it for Asplenium. Here's one we could find, and we'd maybe scoop that up, put it in a petri dish, take it to the lab, and we could see that, yes, these things are, reproducing in nature. So ferns are making tons of spores. Are they reproducing in nature? Yes, they are. And I think here's another one that's got maybe its first leaf that it's made. And here's the gametophyte. Very small, very transparent. You really have to know what you're looking for to find it. But once you get, once you see it, So here's the number of spores that we calculated in Asplenium platyneuron, 2,300,000 spores per frond, which was more than, say, or a better calculation than it makes lots, because nobody had ever figured it out for Asplenium platyneuron. And then Woodsia obtusa, almost 1 million, about 910,000 spores per frond. And it's got a smaller frond, so we would expect that. More species to study. So we've got more species just because we found that in Asplenium and Woodsia. Out at the field station is Thaleptris kunthii and equisetum. There's a species of equisetum out there. We would like to collect some equisetum cones. It has a cone, and do some calculations there, measurements. Also, polypodioides, resurrection fern is there. And over at the museum across the street is Anocleus and Sibyllus, and nobody's ever made any calculations on that. So really, people working on ferns, a lot of people study where they grow, but gametophyte development, not too much work with that. And the more people look at gametophytes, the more they see that they're not all the same. There's quite a bit of differences. So we can tell the difference between Asplenium platyneuron gametophytes in nature and Woodsia obtusa based on hairs that they make. One of them's got hairs like on the surface, the other, they're more on the edge, but they're a different shape than what anemia has. Anyway, I think that's all I could, I'd be glad to answer any questions. I've worked with ferns Well, I added some, I looked for a compound. I did a lot of chemistry many, many years ago, but now my HPLC broke, so I can't do that. So I haven't been doing any of that work lately just because I don't have the equipment. And a collaborator in Australia, he retired and he's probably 80 something now. So anyway, so I kind of have abandoned that, but somebody might have thought, why were some gametophytes female, some gametophytes male? It all has to do with the hormone, plant hormone. They make a hormone, and that's what I studied for many years, answering the question, what is the chemical structure of that hormone? And we made some progress, but there's still more progress to be made. But I'm going to come back over here. Anybody have a question? So it's not even 6 o'clock yet. You guys can go and start studying chemistry or biology or whatever. I know biology people always have stuff to study.
I had a question for you.
Yes.
The lateral meristems. Do you know of any researchers, or do you yourself have any plans to do stuff like gene expression or anything molecular involved with like why the development of the lateral meristem?
People, I don't think anybody's there. All the molecular work is done with sporophytes. I don't think anybody does any molecular work that I'm aware of with gametophytes. I think it's just something that people haven't done. People just, so people, even the just the development, and it's just people, there were people studying development years ago, but most of those people have passed on or died. So it's just a field that mostly the people look at sporophytes. Yeah. So if somebody wanted to do some kind of molecular work or something, yeah, gene expression, it's wide open field.
I say relatively easy. I mean, that probably would.
The problem years ago was quantity, or how much. Now, I mean, technology has changed a lot. So, so anyway, there's also a student. There's a hybrid over at the museum of Philipterus, and I had a student but this was really a lot way before even some of the DNA technology and things that were going on. But we theorized that it was, and looked at morphology between what we thought were the parents. But her skill set was not molecular, and my skill set, I could do some molecular, but not working. I was more working with proteins and chemicals as opposed to DNA. And so DNA technology has, improved tremendously in the last 15, 20 years. So if anybody would like to work on a really cool, isolate DNA and do some sequencing or something between a hybrid and what two parents might be, we never were able to show conclusively that the parents were these two, but morphology seemed to indicate that it was that, because the hybrid was kind of in between. But if anybody's interested in hybrids and doing some DNA work, come see me, because we have material, around. We've got material here. I
think I know a guy who's interested in botany. He says he's interested in botany, so I could ask if he'd be interested in doing it.
Yeah. So if somebody knows those, I'm sure you guys, even in regular old genetics, in your genetics class, are just doing DNA stuff that, I never did. We didn't have it. And then when I taught molecular, I taught more with proteins, and then somebody else came in and did the DNA. And even then, that's been many, many years ago. And like I said, technology has just expanded. Yeah. So you don't need a ton of material like maybe years ago.
All right. Does anyone have any questions?
I had a question, Dr. Hudson. Whenever you said that you kept the little spores in the refrigerator,
Right.
how have you ever done any studies on maybe like the heat resistance? Like, I know spores, they have that big, that thick coat.
Yeah, so, they got that thick coat. Yeah, that's something I haven't done. What I did, I think one of the first few years I was here, there was a high school student that wanted to do a project. So I'm not sure if he wanted to really work on a project or just get out of class. But anyway, he came over And we took spores and put them on plates. This is back in the 90s. We put them on plates, and then in micro, there was a UV lamp, and he put them under the UV lamp for various amounts of time, and then looked to see if they grew, and they actually all grew.
Wow.
The UV light did not do a thing to them. It was like, well, the UV light was supposed to kill bacteria. And I mean, I wouldn't have put my hand under those UV lights, but he was like, put them under, and then it would be a very simple experiment, but I think they all grew. We never could keep them under there long enough to kill the spore.
Wow.
From what I remember. So, but anyway, that's where, but heat, I don't know. You know, that's a good question. But I've always kept my cold, so I can't do a heat study.
Yeah.
I could start now, and then when I'm much older, I could check it out.
Yeah, I kind of had another question too.
Yeah.
Did you ever fully grow those ones that you had from 1978, the ferns? Like, were they fully formed and perfect and everything? All the leaves were fine and everything?
No, well, that's where this particular fern, Anemia mexicana, it grows in the hill country, and it's usually on a vertical surface, on like a road cut or a side of a cliff, something like that. And so I have never been able to grow that plant in the greenhouse. So if I get sporophytes forming, for years and years, I would get sporophytes forming. They would grow. I would transplant them. Now, I never could mimic that place where they were out in nature. Never could do that. The gametophytes grow fine on those plates, but I never could mimic that. Now, there was another anemia species, Anemia phyllidus, that I worked on at Iowa State for my graduate degree. I compared the two, or I isolated hormone from it and Anemia mexicana. And that one I grew I was able to grow sporophytes, and they produced spores, but mexicana, I tried and tried, and I never could mimic it. But when out in nature, I collected along road cuts, like along the side of the hills, and like kind of vertical surfaces along by the Frio River, or where I get my spores. And it's never growing on a flat area, hardly. Yeah, but good question. But I can get sporophytes, but I can't really grow those sporophytes, because I just love gametophytes.
Yeah, it's an interesting study. Yeah. Thank you.
Yeah, good question, but thanks. So you guys, time to say goodbye, and you guys go do some studying. If you know anybody that wants a simple project, or likes to take pictures, and or likes to do a lot of counting and keep track of numbers, how many spores are produced by a fern leaf? It's like nobody's figured it out in a very scientific way, other than, oh, it's a lot. We need more than just that.
All right. Well, if no one else has any more questions, thank you, Dr. Hudson,
Okay. All
for
right.
presentation.
Or let me go stop sharing. Let me go stop sharing. Okay. There we go. Okay. So you guys do research. It doesn't always work out. This leads to that. You never know where you're going to end up. Yeah. And if you want to, I'll keep Tyler posted with the field station. That field station is a wonderful place to do things too. So anyway, okay, I'm going to leave. Bye, everybody.
All right. Thank you, Dr. Hudson. Okay. And see you around.
See you around.